This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 27, recorded on April 11th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is the podcast all about parasitism. And joining me, as usual, is Dickson Despommier.
Hey, Vince.
It's 4/11/11. How about that?
Wow.
It happens.
It does.
This year it will be 11/11/11. Maybe we should do a TWiP on that day.
Oh,
also.
we have to do that.
How have you been, Dixon? I haven't seen you since Friday.
It's been fine.
And it's Tuesday. No, it's Monday, isn't it?
It's Monday.
We record TWiP in a stochastic manner.
We do.
You do know what I mean by that, right?
I do.
Excellent. You are a scientist.
Yeah.
You seem subdued, Dixon. Maybe Monday is the good day to record TWiP.
It's possible. Why, would you prefer me subdued rather than animated?
No, you just be yourself.
I'll try my hardest.
I saw this movie this weekend called The Tourist.
Oh.
With
Johnny Depp.
Johnny Depp and
Angelina Jolie.
And he says to her, "Would you prefer that I do it this way?" And she says, "I just want you to be a man and do what you want."
Whoa.
Just do what you want, Dixon.
Okay.
Which includes picking the parasite. And
Bingo.
You know what? I sat down with you today,
Yeah.
and I don't know what we're going to talk about. I've
know
got
you
your
don't.
book open
You do.
to the table of contents,
You do.
and I'm going to know in a few moments what we're going to talk about. Last time we talked about the one spread in snails. Wait a minute. Schistosoma monsoni. And schistosoma something else.
No fair reading, Vince.
Hang on. I always say that you don't need to know it all. You just need to know where to look for it.
Now, this is all true.
And when it goes to parasites,
Yep.
it's a trematode,
It is.
Schistosoma japonicum,
Yep.
hematobium,
Yep.
and mekongi. I do believe we mentioned all four.
We did.
Those are trematodes. We've left the nematodes.
We've barely touched the surface of the information of those
Yes, we did.
wonderful blood flukes also, but we're going to move on to some other trematodes today.
Good.
In fact, that's how they're classified. Other trematodes.
Really?
Yeah, I'm not kidding.
Are they trematodes of minor medical importance?
Well, you know what? It depends on where you live.
Yes.
If you live in the United States, of course, the answer is definitely minor, rarely occurring, and often never thought of by clinicians with regards to disease.
That's what we're talking about today?
We are, but if you live in, let's say, Korea, south or north, doesn't matter, China, Vietnam, Southeast Asia in general, these are major infections. These are major infections.
And they are also trematodes.
They're all trematodes, is that we're going to talk about today, and they're all snail dependent.
Okay. All of them transmitted by snails, like
not transmitted by
schistosoma.
snails. No, they're not transmitted by snails. Just like the snail is not the vector of transmission for schistosomes.
Yeah, it was just a place where it grew.
It's an intermediate host, Vince. It's called an intermediate host because
if I remember, they get out of the snails and then they burrow into you, right?
They swim.
Yeah,
they do. In fact, all of these trematodes require fresh water for their transmission.
And an intermediate host.
At least one.
Which is the snail, right, for all of them?
That's correct. Not the snail. There are many varieties of snails. Each one is dependent upon a separate species. Amazing. All right. So why don't you ask me a question here? Vince? You've got my book open. You've got it open to Clonorchus sinensis.
Clonorchus sinensis.
Which is only one of them.
Is that one of the ones we're going to talk about?
Yeah, sure it is.
I mean, that follows the chapter on schistosomes.
Yeah, that's right. And so when we wrote this book, I should say, and we'll be rewriting this book, we wrote it for an American medical student audience.
Is that right?
It is right. So we've tried to prioritize the subjects so that they resonate with the residents. and the medical students. So.
Are you thinking of your Alan Dove today? They resonate with the residents?
Yeah, well, it does rub off.
This is a provincial textbook, in other words.
It is. It's culture bound.
Do your medical students here, did they used to buy the book?
They did.
It was part of the course, right?
It was. It was highly recommended, but not required, of course, because you can't really promote yourself like that.
You should make an electronic version.
I've been thinking about doing that, actually. It's not so easy, though, because a lot of electronic versions of books don't allow the pictures to be included.
Well, they're color. That's the problem, right?
Well, it doesn't have to be color, but they do have to be included.
I think color enhances it significantly.
Of course it does.
So I think you should make sure you have color.
I will.
We are working. I say that tongue-in-cheek, as you know, because we are working on it.
This is true.
So, Clonorchus sinensis.
No, let's back up a minute.
We want to back up?
I do, because this book will contain a small sampling of flukes. They're all called flukes.
Small sampling, as if we're doing a tasting menu, right?
Well, I wouldn't do one for this one if I were you, Vince, especially when it came to raw freshwater fish, because that's exactly how some of these are transmitted, but not all of them. If you look at a checklist of all of the infectious diseases found in humans that are transmitted through the ingestion of food,
right,
the flukes would by far top the list in terms of numbers of species. They would far outnumber the bacteria, the viruses, the protozoans, and other helminths. There are by far more flukes out there waiting for you to take a big bite of something.
I cannot resist. So it's not just a fluke, right?
It's not just a fluke. And thank you, Alan, again, for sharing your earworm with us.
Earworm?
Earworm. You know what an earworm is?
Is it really go into your ear?
No, it's a thing that you hear and then it keeps repeating again and again in your head, like a song that you want to get rid of or a jingle.
So in terms of the different kinds of flukes, these guys win. But in the terms of the number of hosts infected, Trichinella probably wins, right? Foodborne, that is. You
mean in terms of the total number of people infected by a pathogen? Yeah, these are not... Unless you live in a specific area where they're highly endemic and it involves one of their national food dishes, then you're right. But about 40 to 50 million people throughout Southeast Asia harbor these infections.
The various ones we're going to talk about today, in addition to schistosomes.
Yeah, that's right. And in addition to that, there's about a 750 million to 1 billion people at risk because they eat the same foodstuffs that these other people ate to become infected. So I'll tell you some trends, and it's a trend. A lot of trends are in vogue right now, which aids and abets the spread of these infections that we'll be talking about. And one of the big ones is fish farming. Freshwater fish farming is on the rise in many, many different places throughout the world.
Freshwater
fish farming.
Now, we have a huge saltwater farming industry, right?
We do.
So you're saying that fresh is starting to ramp up?
Well, if you live in Southeast Asia, they eat a lot of freshwater fish, carp, or cyprinoids as they're called.
Mm-hmm.
So the demand is going up because the population is rising, right? So as a result, the fish farmers increase their yield by increasing the amount of land dedicated to fish farming. So a lot of these parasites, in fact, all of these parasites depend on human fecal contamination. Every one of them. So this is a good indicator as to the use of human feces.
So these fish farms often get contaminated?
Often. I mean, imagine yourself as a fish farmer. You're far away from your house. You're in the middle of this waist or knee-deep fish farm, and you have to go to the bathroom.
Right. So you go to the bathroom. You get contamination on your hands, which
Number one isn't
inevitable.
So bad. Number two.
Yeah. It's inevitable to get contamination, as you've told me before.
It is inevitable.
Because the process is inherently defective.
Well, we'll go into that much, much later. That's another podcast entirely. But
is that right?
Yeah, I think so. I think a redesign of the bathroom, of the toilet, is in order here because we can prevent a lot of this simply by sanitization.
But the fish farmer contaminates his or her hands and then goes back to work.
Not hands. No, they just drop trow, as it were, right in the middle of the fish farm or on the edge. Are you
serious?
Of course.
I thought here they're going to a bathroom and not washing their hands properly.
No.
But they're actually defecating Not into the waters
they
where
know,
the fish, but nearby, right?
Vince, they know from experience that human feces is a good fertilizer because that's what it's used for the crops on the land. So if these fish are eating plants that grow in the farm pond,
right.
well, then a little bit of human feces goes a long way.
So they defecate right into the waters where the fish are growing?
They do, but there is another part to this, of course, that's only fair to mention, and that is a lot of them have reservoir hosts. In fact, most of them have reservoir hosts. In fact, if I think carefully about this, they probably all do. Mm-hmm. There are a lot of these intestinal flukes, as they're referred to, but a lot of them don't live in the intestines. Some of them live in your lungs. Some live in your liver. Some live in your bile duct. Some live in the gallbladder itself. Mm-hmm. These flukes infect a wide variety of animals, not just humans. So even if humans stopped defecating ad libitum and employed a proper sanitary facility, the animals that run wild around here could still...
Squirrels, foxes,
not
whatever.
Squirrels, but these has to be carnivores, like cats.
Dogs.
Yeah.
So these are around the fish farms.
Sure.
So a fish farm has large bodies of water. And
crops growing next to it also. They're not just single entities.
Really?
Yeah, and a lot of them have fowl, like waterfowl, like ducks.
Dickson, if you did this in a building, say a skyscraper, maybe you could get around the fecal...
maybe you could, Vince.
That's not a bad idea, is it?
We should try to develop that somehow. What would we call it?
Can you put a fish farm in a vertical farm?
Of course.
And then you don't get the contamination from the carnivores that are defecating into the water and putting flukes in it.
Yeah,
right?
A lot of this stuff is preventable by design.
Do we have freshwater fish farms in the U.S.?
Sure.
Are they subject to the
catfish.
same...
Every time you go to the store and buy catfish.
Right. Do they get contaminated with flukes?
Not in this country, primarily because we don't have the proper snail hosts.
All right. The snail is essential, in other words, for all of these.
Not just a snail, a proper snail.
A proper snail is essential.
Yes, of course. We only eat the proper snails,
Okay. We don't have the right species, in other words.
Yeah, right? You have to have the right species.
Okay.
And a lot of these are coevolutionary trends, and you can watch the snail develop in the parasite and the host at the same time. And they're linked. Mm-hmm. All of them are linked together by common habits.
Now, why are these flukes in this book if it's for U.S. medical students?
Because sometimes they travel. And sometimes people travel from there to here.
And they're sick and they come for care here.
Right.
Okay.
Right. So let's just talk about them in general first, and then we can talk about a few specific examples.
Okay.
Because I think it's important to just nail down in the mind of the listener right now how widespread these things are and how you catch them. Okay. So I would say to you, as a ingester of comestibles, Vince, how many raw fish did you eat last week?
Actually, we did have sushi Saturday night, which is unusual. It's the only time.
Sushi is a freshwater or saltwater fish type meal? Think carefully.
Think carefully? It depends.
What if I gave you just two choices?
No, you don't have to do that because I think most of the species there were saltwater.
Correct, except for smoked eel.
It's usually cooked, though.
It is. So you're safe there as well.
Yeah, I don't think we had smoked eel. It's one of my favorites, though.
Me too. And they have shrimp.
A lot of shrimp. Salmon is very typical.
Shrimp is cooked, though. But the salmon is not cooked.
And I had some without any fish whatsoever.
So uncooked salmon on the West Coast has actually transmitted one of these flukes to humans.
Because they spend some time in fresh water?
Correct. They're anandromous.
How do you spell that?
A-N-A... A-N-A-N, anandromous.
D-R-O-M-U-S?
Something like that.
Which means the fish goes from fresh to salt?
It lives as an adult in salt water,
right,
and spawns in fresh water.
So in the fresh water, it picks up the fluke.
Yeah, but eels are contra-andromous. They live in fresh water, but they spawn in salt water.
So you said some fraction of the salmon grown on the west coast of the U.S.
Not grown. Captured wild
salmon.
Yeah.
If you farm salmon, you keep them in salt water the entire time, right?
This is true. Yeah, they don't have a chance to come back and spawn.
So that's not a necessary part of spawning, being in fresh water?
If you have a fish hatchery, you have to the eggs have to hatch in fresh water. The smolt and the parr have to develop in fresh water.
So a salmon farm has to have a freshwater component.
It does.
And do you do that on land? Because I know a lot of the salmon farms are offshore, right?
You probably do it along a river.
Okay.
Okay, and you divert part of the river into the salmon farm to raise the fish to a certain size before they're transported.
And then you bring them out to your offshore.
Gradually.
This has become This Week in Fisheries. But I think this is fascinating to know this. All right, so the farmed salmon on the West Coast don't have flukes.
Not the farmed salmon. That's right, because they never get a chance to come back.
But right. But the wild ones. So can you buy wild salmon?
You can.
And how would I know? Because I don't want to buy it anymore.
It's sold.
It says wild salmon.
Some of it is quite abundant.
I don't want liver flukes. No, not liver. I don't want flukes, period.
You could also get the fish tapeworm that way too, but rare, very rare. In fact, that's not a pun, but indeed, you'd only get it if you ate it raw. Which is why a lot of people wouldn't encounter this, because most people don't like raw fish. They prefer their fish cooked. Even the salmon that you buy at the store, even though they recommend only cooking it for a certain length of time, the majority of people living in this country, at least, prefer it cooked all the way through.
Do you?
No, actually.
You like it raw?
Not raw, but a little bit on the rare side.
But in Asia, people love raw fish, right?
They live on it.
And that's why this is an issue.
Now, why is that, Vince?
Why do they like raw fish?
Yeah. Because cooking requires energy.
I was thinking of that, but in the old days, they had fires, right?
Oh, sure, and they still do, and they still cook their rice this way. You can't eat rice unless you cook it right.
Well, it's become a custom. Now they like it.
It has become a custom. That's right. And you can eat a raw fish anytime, anywhere, without having to worry about cooking it.
You know, when I was a kid, we used to go fishing out in the ocean a lot.
Yeah.
And I remember once there were some Korean gentlemen on the boat with us, a party boat, and we were catching bluefish mainly, which is what you do off the coast of Jersey. Yep. And the Korean gentleman would come up to you and say, "May I take the cheeks?" And they would cut the cheeks out, and they had a little bottle of vinegar with them. And they would put on, boom, they said it's the best part of the fish, the cheek.
You know, it's ironic that you should say that, because my kids were raised with that concept, because the Romans, when they invaded some places like the British Isles, for instance, and Spain, would catch salmon, all right? And they would cut the heads off and throw the bodies of the salmon to their captors.
Oh, my gosh.
And they would only eat the cheek muscles. Only.
Wow.
So my kids have immediately glommed onto that concept. So we had to always save the cheek muscles for them.
But you could get a fluke from the cheek muscle, right?
Sure. Well, we'll get into it, because in Clamorcus, it's not the flesh of the fish that you're going to eat to get it. It lies just under the scales. So if you eat this fish whole with the scales, imagine eating a fish with scales.
You don't eat scales, do you?
Some people do.
But they're kind of scaly.
You mean hard and bony?
Yeah. Don't you scrape them off?
Yeah, most of the time you scale the fish, but with bluefish.
You eat the skin.
Yeah.
I don't remember, but they must have a light
And with
scale.
trout, it's the same with trout and salmon. They have very, very small scales. There are 4 kinds of scales, Vince. Did you know that?
Nope.
Yep. The most primitive scales.
I know the A scale, the minor, F minor. Sorry.
No. Okay, so there's the ganoid scales, right, on primitive fish like sturgeon. You can see these almost like plates, like bony plates along the outside of the fish. Those are called ganoid scales. Then you have even more primitive scales. Before there were teleosts, there were chondrichthyes. And what is a chondrichthyes?
A shark?
You see, you shook your head no, but you knew the answer. You know what kind of scales they have?
No.
Placoid.
Placoid?
Placoid scales.
Wow.
And they all point backwards. They look like little hooks. But they're so small that you can't see them.
If you rub your hand the wrong way, it hurts, right?
Exactly. Well, not it doesn't hurt, but you can feel it. It's like sandpaper. Exactly right. Exactly right. So I often said, how can you possibly wear a sharkskin suit? It was probably colored the same color as a shark skin, but not made out of shark skin, right? And then there's cycloid scales. Cycloid scales are in most teleosts. Those are the kind that you would scale off, let's say, a porgy. Or a sea bass, if you caught them in the ocean. And then there are these tiny, 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 tenoid scales. Ctenoids.
Mhm.
And those are the kind you'd find on salmonids, which are a more recent teleost in terms of evolution.
And the parasites live just beneath.
They live underneath there. They live right underneath. Why is that?
You have a nice photo here in your book.
I do have a nice photo of it, and it's because of the way these things acquire them to begin with.
All right.
So the life cycle is pretty... Let's start with a basic life cycle,
Any
okay?
particular one, or
Doesn't
they're all
matter.
the same?
Doesn't matter.
Before you do that,
Sure.
just run off some names. Besides,
you want some names from me?
Besides Clonorchus sinensis.
You have Metagonomous yokogawi.
Wonderful.
You like it?
Metagonomous yokogawai.
Or yokogawi.
Okay, go ahead.
Heterophes. That's an easy one to remember.
Okay.
Right? Paragonomous westermani.
It's not even in your book. Oh, it's
Sure
in
is.
another chapter. Yeah,
It's
okay.
there because it's not a minor parasite. It's a
Okay.
pretty serious one. Those are just a few. Fasciola hepatica. Fasciolopsis.
Echinostoma.
Fasciolopsis busci.
How about heterophytes? Oh, you said that. How about Nanophytes?
That's the one you get from eating West Coast salmon.
Nanophytes salmonicola. No,
Salmonicola. It's a rare parasite. That is to say, it's rarely occurring as a clinical entity. Okay, but it has been recorded as an
you'll
infection.
tell us in time what the symptoms would be of an infestation.
Well, some of these are quite serious, like Paragonomous, for instance, that you get from eating. What, Vince? What could you eat that you could catch Paragonomous from?
I don't know
A
Paragonomous.
raw crab. A drunken crab. It's an actual meal in some areas of Southeast Asia.
I'm just thinking,
crab.
we don't eat raw crab here. We definitely cook them, right?
So how would you catch these freshwater crabs? Tons of freshwater crabs all over the world.
Large?
Some of them.
Okay.
They're about yay big. Okay, I'm going maybe the size of a softball altogether, right? And you throw them in some marinade, and it's got some alcohol and some vinegar and some spices, and of course the crab hates it, but it doesn't die. It just sort of gets drunk.
Kidding. That's why they call it drunken crab?
Yeah. So when they take it out of the marinade and they put it down on the plate, it doesn't walk away. It sort of twitches. They crack off a limb, suck out.
This is barbaric.
Isn't it?
Well, we do a lot of barbaric
Come
things.
on, you ever go down south and eat crayfish?
No.
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 You boil them up first, which saves
Right,
us
you
from
kill
a
'em.
lot of things, right? In some Creole spices, and then you twist off the heads,
Yeah, I don't like
and you
that.
eat the tail, which is like lobster.
Mm-hmm.
But then they say the best part is the head itself. You suck out the heads.
Do you do that?
I've done it.
Since you're from New Orleans?
I've done it.
All right. So,
with enough cold beer, those are delicious.
the drunken,
the drunken crab dish is a Southeast Asian dish, and
what can you get from that?
Paragonomous westermani.
And this could be fatal.
With enough of them, they live in your lungs, and they get to be the size of a small. They're a little bit bigger than your thumbnail.
All right. Is the life cycle the same for all of them?
Yeah, basically it's the same.
Let's do it.
Fine. So somehow the eggs of these parasites have to exit. So
Exit us?
Yeah. So the parasite is living somewhere inside of us. Okay, the liver flukes are living in the liver. The bile flukes are living in the bile.
They live in many places,
the
as you
lung
said
flukes
earlier.
are living in the lung. So wherever they are, those eggs.
Each one has a specific place where it lives.
This is correct. They site select for a niche.
Okay.
So they're living there as adults, and they've mated. Okay, now just like the schistosomes, all right, the trematodes, many of them, not all of them, but many of them are hermaphroditic in the sense that they can fertilize their own eggs, but some of them need to pair off with another to exchange gametes.
I think schistosomes had to do that, right?
Yeah, but no, they live as a male and female worm, and there they are. The male and females are distinct. They're not hermaphroditic. There's a male and there's a female.
But you need at least two.
Yeah, but in flukes, they have both male and female organs.
Okay, so you just
you'd
need
think,
one?
Well, no, you see, that's what I'm driving at. Even though they have both sex organs, they still need to cross-fertilize.
Okay.
Snails are the same, by the way. Do you ever see this movie? I'm going to pick this movie again. I think I picked it once on TWiP, but I think I'm going to pick it again on TWiP. It's called Microcosmos.
Yeah, I believe you did.
Right. And in there's a scene where two snails come together, and they play some kind of a Debussy melody to get you into a romantic mood because it's like mating snails. Well, snails are hermaphroditic also. So here we have hermaphroditic parasites infecting hermaphroditic intermediate hosts.
Mm-hmm. Okay.
So the eggs pass into the environment, which means that they have
feces,
to get
right?
They have to pass, not always.
Mm-hmm.
No, with a lung fluke, you can cough them up and throw them out in your sputum. Right? All right. But for the most part, it's intestinal tract. Even there, you can swallow the sputum, and the eggs have to... Eventually, they exit the body. And they have to be deposited in fresh water in order to continue the cycle. The eggs, then, all different in shape and size and form, contain a similar life cycle stage inside, called the myricydium. The myricydium is the same stage it was in the schistosome eggs. This little swimming ciliated stage, it's free living. They're free living, Vince.
I'm looking eggs on your picture here. Eggs, myricydium.
And the myricydium...
And then it goes into a snail.
It has to find the proper snail. The proper snail. So it's not just any snail, it's the right snail species. And each one of these flukes has a separate snail host. And you might imagine somewhere way back when there were only invertebrates alive in the world, this might have been a parasite just of snails. And somehow it evolved into a parasite of vertebrates, cold-blooded vertebrates first, and then finally warm-blooded vertebrates.
Yeah, it encountered new hosts. It evolved. It adapted.
Successful systems attract parasites. So here we have the egg going into the environment and hatching, releasing its myricydium, which then swims around.
Just in the water, right.
Right, and imagine how big an environment this is for this parasite.
Huge.
And yet, Vince,
it finds a snail.
How does it do that?
Smells at chemical cues. Absolutely.
Amazing. Now, we just lost a brilliant supporter of that database, and that was Thomas Eisner. He studied pheromonal attractions in insects and things like this, and in arthropods, but I know that he was attracted to these kinds of subjects regardless of the phylogenetic level of discussion. So here we have a very, very, what would look like a primitive invertebrate, right? A fluke stage seeking out and finding its snail host.
Right.
Bang. There it is right over there.
All
okay,
right.
so fine.
What does it do when it finds it?
Penetrates the foot of the snail.
Oh, it says here the eggs are eaten by a snail sometimes. Is that possible too?
It is possible. But I mean, we're giving you the general life cycle here. But you're right. In many cases, the parasite would be eaten first.
And then hatch.
So in that case, the snail finds the egg rather than the egg finding the snail. So the snail is a detritus feeder. Feces is part of the detritus scene.
On the bottom of the water, whatever it is, lake.
Let's make it a fish farm.
And the eggs sink to the bottom?
Well, the feces is there, and then it sort of...
Sinks, yeah.
Well, feces actually doesn't sink
to
begin with, no. Feces actually floats because it's got lots of air pockets in it. Any kid that's swum in a local swimming pool will tell you
Dickson,
that one.
I got to tell you, it doesn't always float.
No, it depends on the kind of feces that you're expelling.
Okay, I don't mean to be crude.
No. We're not being crude and rude. We're just going to tell it like it is. And if you live in Southeast Asia, people have 3 or 4 or 5 bowel movements per day.
Why is that?
Because they have soft, diarrheic-like stool.
Because they're ill?
Not necessarily, but there is something going on there that doesn't go on in our gut tracts. We usually have a single formed stool per day.
This week in stool, huh?
Well, it's it might be stool in quotes to you, but it's a lot of people's bread and butter because that's what they look at in order to make their living.
Yeah, bread and butter, Dickson.
Well, that's what they say. It's their funny little expression. It might be blank to you, but it's my bread and butter.
So if it's not well formed, it floats.
Or it dissipates.
But if it's formed, it would sink, right?
Yep. So along comes the snail. The eggs eventually do fall down onto the bottom, and the snails
would eat it. Okay,
but
eat it. The alternative is that the
myricydium hatches and then
burrows into the foot of the snail.
That's right. And there are plenty of examples of both of these things. Of course, with schistosomes, it was the swimming myricydium. But in the case of most of these other flukes, you're right. The snail does the ingesting part. But that's too bad for the snail because what happens next is basically the same thing that happens with schistosomes. The parasite undergoes a rapid transformation into something called a sporocyst.
Mm-hmm.
And the sporocyst is a bizarre developmental stage of this parasite that produces offspring. And that's why they're called daughter sporocysts. And the sporocysts are, they're just a weird form. If you take a look at their biology inside the hepatopancreas of the snail, you don't know what you're looking at.
This is where they go into the hepatopancreas?
They do. It's like looking under the electron microscope at a virus factory inside the cell itself, only on a macro scale. And it's in various stages of development, and it's not synchronous, and it's producing them all the time. So a snail could be productive for most of its lifespan.
Doesn't injure the snail?
Well, enough of them would.
Mm-hmm.
But a couple, maybe not. But they produce thousands of offspring.
So it's making more Miracidia? No.
No.
Miracidium is the egg.
No. Miracidium is inside the egg. Miracidium is the swimming stage inside the egg. But in this case, this Miracidium doesn't have to swim, does it?
No, it's in an egg, yeah.
Okay.
But if it were a swimming form, it would just be a Miracidium.
That's
right?
correct. So
now, in the snail, what is it reproducing into what? More Miracidia?
No. Into a sporocyst stage first, and then into a daughter sporocyst stage next, and then the daughter sporocysts give rise to the... You're sitting right there reading it?
Cercaria?
Bingo. And
they have the tails, they can swim.
Yeah, and those are the same forms that the schistosomes.
So there's a form here with a lot of cilia around it.
Yeah, that's the Miracidium.
That comes out of the original egg. So then these Cercaria leave the snail.
They do.
And they can swim in the water.
They can.
Okay.
So in the case of the schistosomes, as you'll recall, the Cercaria were following a chemical trail that we were leaving on the surface of the water. Right. Something like lanolin and other oils in our skin that we produce in our sebaceous glands. And these Cercaria are positively phototropic, so they come up to the surface of the water, and now they can sort of look around with their chemical receptors to see where the strongest signal is coming from. And once they find that strong signal, they start to swim towards it.
What signal are they looking for here?
In this one?
Yeah.
It varies with each one of these flukes now. And then that's where the food items come in.
Mm-hmm.
Okay, and some, they're not even looking for a chemical trail. They're just looking for a piece of vegetation. Let's talk first about the one that most kids know in biology in this country. It's called Fasciola hepatica.
Yeah, you would learn about this in biology?
You wouldn't learn all about it, but it's an example of a fluke, an invertebrate that you can actually see with your naked eye. And they're beautiful, actually. When you stain them up, they're gorgeous. They're sinisterly gorgeous.
Fasciola hepatica?
Fasciola hepatica, the sheep liver fluke. And they're so common, even in this country in certain places, that they're easy to get for biological supply houses. So
I see.
They can find them all over the place. And they're huge. Like I said, they're the size of a small leaf on a plant.
Would that be it?
No.
That's a Cercaria.
It is.
It's not the adult.
No.
Yeah, you don't have any adult photos here.
I have one of a Fasciolopsis, I believe.
You have a...
there it is.
Is that it?
Yeah.
That doesn't look beautiful to me.
Oh, it is to me. It's gorgeous. When you look at these things under the light microscope, and you've seen a perfectly spread out one with the organs all inside and everything, they're gorgeous.
30 millimeters by 14. This is large. And this is living in your liver?
It's living in your liver. So let's talk about what the Cercaria does afterwards.
So the Cercaria are swimming. They find food. They head towards food, right?
Well, not necessarily. You see, for it varies. Fasciola hepatica, they will go to the littoral vegetation.
Watercress.
Littoral.
What does littoral mean?
Littoral means, it's an ecological term. It means the edge of the lake.
Is that like an ecotone?
It is, Vince.
It's kind of an ecotone, right?
It is the ecotone between land and water in fresh water.
You see, I listen to you, Dickson.
Not only do you listen to me, Vince, you remember, and you apply them properly. So that's a brilliant statement from you.
All right. So the littoral has, in your picture here, they have watercress.
But that's not the only thing that grows along the side of a lake. And so these Cercariae will seek out vegetation along the side of the lake, and then they do something quite magical. They attach with their sucker disc. Their tail falls off.
Attached to part of the plant that's below the water, right?
Yes.
Okay.
And they round up, and they secrete an impervious shell, a transparent impervious shell over them, and they become the meta-Cercaria.
Wow.
It's an environmentally resistant stage which can last a long time.
This is the one that attaches to plants, right?
In this case, in Fasciola, correct, but there are others that attach to fish, some attach to crabs.
Attached to fish. So when we talk about them being under the
they're not parasites
scales.
of these. So the fish is not a host.
Yeah, these are just attaching.
The fish is a host. It's a very special kind of host, and I think I mentioned this in like the first time we talked about this. They're called paratenic hosts.
I see, because the parasite doesn't multiply in them
or do anything else. It just sits on it.
So
are there any viruses like that, Vince?
Temporarily.
What?
So when you're infected with HIV, it attaches to dendritic cells, but it doesn't replicate in them.
No, but I'm thinking of something more generic, like smallpox. It could attach to blankets, for instance.
Sure, for fomite transmission.
Fomite transmission.
Yeah, sure.
So a fomite is an inanimate object that serves as a carrier for a disease from one person to another.
That's right.
And a paratenic host is a fomite. It's a living fomite.
It's a living fomite. Okay.
It is what it is. It's a living fomite. So you've got living fomites for all kinds of flukes.
So,
and many of them turn out to be food items.
The parasite gets under the scales somehow.
Yes. Not just somehow. The scales, they overlap. They're like shingles.
Yeah.
On
a house. It just slides in
between.
And
they're attached at the base to the skin. Okay, so it slides up underneath and then drops its tail and attaches because this stuff is really hard stuff. If we could find out what that material was that the Metacicaria secreted and you built a house out of it,
it would be sturdy.
You'd have the world's greatest house.
It might smell, though.
No. These are the building materials of invertebrates are incredible.
Yeah, I'm sure we know what it is.
No, we don't.
Why not?
No, we don't.
Nobody has bothered to check it out.
You know what? Go online. I bet you can't find anything on it.
Okay, that's a shame.
Isn't it?
We could learn from this. Right?
No kidding. So we could biomimic it and make things out of it that would be impervious and indestructible.
So tell me how impervious this shell is.
Pretty impervious, all right. You can
heat it
to a point.
Low pH
to a point.
UV.
No problem there. Nothing's dividing, so there's no UV damage of any sort going on. Even if the plant dries out.
Well, you could damage the DNA.
Imagine this, Vince. You know what a water chestnut is, right?
Yeah.
There's a parasite that infects the surface or attaches to the surface of the water chestnut plants. All right? And if you don't eat the surface of the plant, and no one does, you want the chestnut inside, you're okay. Now, guess what? There are people out there
who like the surface.
No. Their job is to crack open the nuts and save the water chestnuts for the commercial. And they use their teeth to do it.
Why do they use their teeth? They get a knife.
They cut the fingers off.
It's ridiculous. But you would think
That's how they acquire.
They would also contaminate the meat with...
Fasciolopsis busci is acquired that way.
But you could contaminate the meat, no?
Not really. These things are glued to the surface of things.
They're not going to fall off, right?
No, of course not. Their
so
job
why do they get it when they crack it with their teeth then?
Because they swallow a little bit of this stuff.
Oh.
And it dissolves away in the stomach and releases the Metacicaria, which we're getting to the next stage of this infection by now. So the Metacicaria in some way become eaten. None of them can on their own infect you.
So you eat a fish or a chestnut or a watercress? Watercress.
No, yeah, watercress. Right, Vince. Now you got it. Because Fasciola hepatica, in this case, is called the sheep liver fluke. Wherever you find sheep, you will find this liver fluke.
Where do you find watercress?
Around every pond set aside for the sheep to have fresh water.
Only sheep ponds?
No, it's found on a lot of other
where
places.
You go fishing, are there watercress there?
Of course.
Okay.
Of course.
Because people like to eat watercress,
They love watercress sandwiches and cucumber with a little...
But isn't it farmed, though, for our sandwiches?
It is. Okay. But a lot of it is also wild harvested, especially if you go to Ireland. Oh, if you go to Ireland.
How about in Asia? Will they wild harvest the watercress?
They don't have a lot of watercress.
Are there flukes in Ireland?
There are, Vince.
There are.
And some tourists that went over there strictly on an innocent basis ordered themselves up a nice ale and some watercress sandwiches and came back with a heavy fluke infection.
Is that right? It happens.
Yeah.
Wow.
It happens all over the world. So wherever you have fluke, or wherever you have sheep raising, you have this fluke also because it's been spread. Now, you kind of ask yourself the question, though. Wait a minute. I thought you said the snails were specific for these things, okay?
Yeah.
So how can you find this everywhere if the snails are specific? And so therefore, they must have shipped these plants around the world, too, to sort of farm them. And the eggs of the snails were there, too. So a lot of snails have been shipped around the world this way. And birds also pick them up on their feet inadvertently and move them from place to place.
And
so
snails have no problem living all over the place.
Hell no. As long as you give them proper water and food, they're in great shape. So you can see how bioinvasion occurs.
Yeah.
And how these medical problems arise as the result of that, too.
I used to have aquaria, freshwater aquaria.
Yeah, me too.
Buy a new fish. If it had a little snail in it, boom, in a couple of weeks, your aquarium was overrun with snails.
Tons of snails. That's right. So you can't avoid them because the eggs can't be seen, and all of a sudden but you want a few.
They do help clean up, right?
I'd rather have a sucker catfish, actually, because I like
those are great.
to watch them.
Those are great.
Because they would scrape with their scrapers on the sides of them.
Yep.
But the point of this discussion that we're having now is not to point out any particular fluke in general, but to talk about this in a generic sense.
Sure.
And so the next stage of the infection for these foodborne fluke infections, foodborne, you don't catch them by drinking water because the metasecarial stages are firmly attached to something.
Yeah, food, it's either fish or vegetable.
Or a crab.
A drunken crab. Anything else?
Yeah, there's a whole bunch of other things out there that people eat because they're hungry.
And if you eat them raw, you're in trouble. But if you cook your food,
bingo,
then you're
killed
all right.
It. Or even if you freeze it.
Okay.
But I've traveled throughout Southeast Asia. I really have, and I've enjoyed it. And it's a pleasure to eat throughout Southeast Asia. You know why? Because it's all fresh. They can't afford the refrigeration, so they have to pick and eat fresh every day. And a lot of them don't have the luxury of a stove. So they eat a lot of unprepared foods. And you can eat a lot of vegetables raw. That's not a problem, right? But when you start eating fish, I draw the line there of freshwater fish, I think. I love sushi. I love sashimi. I love ceviche. But I don't eat freshwater fish ever raw. And, because I guess I know too much. I know enough not to do that. But there are other things you can catch from freshwater fish like that as well, which we've talked a little about before. But when we talked about ascaris, remember, we talked about the cetacean ascarids that we can catch by eating raw fish that way too. Right. So here's a whole bunch of other things that most of the world hasn't paid attention to this because these poor people receive so little medical attention to begin with. No one has really thought much about this as a neglected disease entity, but it's huge. Yeah,
it says here 25 million.
More. Make it, I'd jump that up to 50 now. And
Far East alone, right?
Sure.
1/4 of Chinese immigrants to the U.S. harbor the fluke.
Which one was that? Clonorchus?
Yeah, Clonorchus. Once they're here, they can excrete eggs, right?
Yeah, but
they won't find the right snail.
Or the right fish.
So it has never established itself here in the U.S.
As far as we know, it has not.
It's very interesting.
Yeah.
Now, once we ingest it, what happens next?
It depends on the fluke.
Okay.
It does depend on the fluke. Let's take fasciola, for instance. This fluke is ingested. It goes all the way down to the small intestine. The metasecaria now hatches to become a very small fluke.
Mm-hmm.
It has to get to the liver, Vince.
Always. Well, it depends.
Well, this one has to get to the liver.
Fasciola.
So how does this parasite get to your liver? That's the big question.
It's very small, right?
It's extremely small. It could crawl back up the intestinal tract through the common duct into the liver and live a happy life, but that's not
I'll bet it goes into the blood.
It doesn't even do that.
No,
it just burrows its way through the entire intestinal wall.
Oh, my
goodness.
It gets
to the... Yes, you should see Vince's face right now.
It just goes out of the intestine.
It burrows right through the tissues until it gets out to the...
peritoneal space.
To the adventitia, which is the fascia holding the intestinal tract together. Yeah. And it begins to crawl along the surface of the intestinal tract until it actually encounters the capsule of glycine.
Mm-hmm.
The capsule of glycin is actually the capsule that encompasses the liver.
Mm-hmm.
Let's just make sure I said that correctly. Glycin, Glisson. I believe it's called glycin's capsule.
Glycin's capsule. There it is.
Yep, glycin's capsule. It surrounds the liver.
It's a collagenous sheath covering the external surface of the liver.
Okay, so that memory cell is intact still. So this parasite then actually burrows through the surface of the liver.
Wow.
As a tiny little entity that you wouldn't even feel that happening,
no.
even if you had sensation in your liver, which you don't.
Your intestine either. Your small intestine doesn't, does it?
I believe it does, actually.
No, because if you have those are colon. Your colon doesn't have sensation, right?
Right. But your small intestine does, but your liver does not have pain receptors.
Yeah, no need.
So here's this little fluke, and now it encounters foie gras humano.
It burrows through the membrane.
Yeah, it goes through glycin's capsule.
How long does this take from intestine to liver? We probably don't know.
No, we have a rough idea. It's about a week. Week, maybe 2 weeks. But then the sorry, Vince just ate a piece of raw fish and he doesn't.
No. How many eggs do you need to? Not eggs, metacercariae. How many would you? Just one.
One adult, one metacercaria.
Okay.
Okay, but if in order for this fluke to produce real eggs, it needs two. They need to mate. All right? So they're not totally hermaphroditic. I'm sure I'm going to get emails from people saying, oh, no, but this one is hermaphroditic and can fertilize its own eggs. So it varies with each of these things, and I'm not going to go through all 125 different species of these to tell the listeners.
Make 125 episodes. Just kidding.
If we were being paid for it, I'd consider it. But
Dickson, that's just not the right attitude.
Of course not. But 125, come on, Vince.
It will take a couple of years.
It would take a couple
Wouldn't
of years.
Probably be that interesting.
And no one would. Well, in Asia, they might love it. Okay, so the point is that eventually... in this case, I'm not sure what I say in my book because it was a long time ago since I wrote this, whether Fasciola needs 2 worms in order to complete its life cycle or not. I do know that Paragonimus needs 2, all right, at least, and they live as pairs of worms in the lungs.
It says, for Fasciola, it says self-fertilization.
Fine. In that case, that's exactly what it does.
Okay.
It's a hermaphrodite in every sense of the word.
Where do they fertilize? In the liver?
Well, they live in the liver. That's how they grow up to be that size.
But so there's one metacercariae
equals one of those flukes.
It gets into your liver, and there it mates with itself.
No. It doesn't get in as a metacercaria. It gets in as a small fluke.
A small adult.
Well, you'd call it a juvenile.
Yeah, not an adult. Okay. And then it goes into your liver, and then it can fertilize itself.
It starts eating your tissue.
And making eggs, right?
And burrowing a tunnel in your tissue the size of the worm.
Which can be large, 30
millimeters. Very
millimeters.
large. So you can actually see the tunnels on ultrasound.
You don't feel anything?
Not right away.
Do you get jaundiced?
Not right away. It depends. There's no blockage, so there's no interruption of the production.
And it's just one adult, right?
It's just one adult. And in fact, this adult has a unique habit of occupying the entrance of the bile duct into the liver. It'll sit at the entrance, and it secretes proline, one of the amino acids that has a very unusual structure to it. Right? It's a heterocyclic amino acid, and this parasite produces a lot of proline. And you know what proline turns out to be? A fertilizer for epithelial cells.
Wow.
And here, there's a lawn of epithelium in front of it. And it goes ahead and mows it and then crawls back up into this pouch that it's created for itself just inside the liver and secretes more proline, and back up grows and it mows the lawn, basically. It just keeps mowing this lawn, right? Meanwhile, it's now self-fertilizing, so the eggs start to pass out. And they're large eggs. They're easily identified under the microscope.
Where do they pass out?
They pass through the bile duct, into the small intestine, out through the feces, into the environment.
How do they get from the liver parenchyma to the bile duct? Do they eggs?
Eat their way. No. This parasite has burrowed tunnels that make the connection.
Really?
Really.
The little network of subways.
Really. These tunnels are huge, Vince. These are not little. These are like the channel between France and England.
Here's a section of an adult in the liver.
They're enormous. They're barely. I had to take that one under a dissection microscope.
You took this?
Yeah, I did. I took most of those pictures in there.
Pretty good, Dickson.
Not bad. Not bad. I mean,
it's great.
It's okay.
Where do you get all the specimens?
Harold W. Brown,
Really?
Kathleen Hussey,
Do
Roger
you
Williams.
still have all these slides?
I haven't looked recently to see if I did or not.
Mm-hmm.
Our course has been discontinued here, so I don't know if they kept any of that
There's
stuff.
no parasitology course any longer? It's part of another course?
I don't know.
Okay.
I actually, honestly, don't know.
Okay.
It was sad when I gave up the course, but, you have to move on.
You're still teaching, Dickson.
I am. That's why we're doing this, because this is a substitute for that course. In fact, in that course, they never got this. In fact, they never got this subject.
Really?
Nope.
But it was in the book.
Yes, but they never even heard about this one.
All right, the eggs come out of the liver.
Yep.
Into the intestine.
Yep.
Into your feces, and they go back into the environment.
Yep.
Now, this is a liver fluke, but then there are gallbladder flukes, lung flukes. Let's just say, how do you get from the intestine to the lung? Same way, you burrow?
You betcha. Not only do you have to now go through...
You got a
you
diaphragm.
have to go through the diaphragm, that's right.
Well, they probably can get through the portal there, right?
And these flukes can locate in weird places, Vince, like at the tip of your fingers or in your brain. They're mostly seeking out your lungs, but they can sometimes make a mistake.
They can burrow to your brain?
Yes, they can burrow to your brain.
How would they do that? Up your spinal column?
Ask them. It's a mystery, but they've been found in brain tissue.
Why the tip of your finger?
They've obviously taken a wrong turn. They didn't have MapQuest. I can just see these flukes sitting at their computers and typing out MapQuest. And then I want to go from the liver to the...
Yeah, I see one here.
To the lung.
Paragonimus westermani. This one is in the lung.
It is, mostly.
Oh, look, crabs. This is a crab one.
Yeah, that's the drunken crab.
We'll have to get all these images.
We will. So and guess where it was discovered first as an adult worm? It was discovered in a Bengal tiger.
Just by accident, I
Paragonimus calicati was the name of the parasite that we discovered in tigers.
I see.
And humans can also catch it.
So if enough in your lung or liver, if enough of these adults are there and burrowing, eventually you're going to get problems, right?
Yes.
But you could live for many years producing eggs?
Many years.
So these millions of individuals in Asia who are infected are...
They're not just temporarily infected. They're chronically infected for most of their lives.
And they're producing eggs, and some of them will die as a consequence of the infection?
Well, that's interesting. Will they die from the infection, or will they die from something associated with the infection? Mm-hmm. I'll give you the example here that we've mostly paid attention to, and that's this smaller worm called Clonorchus sinensis. Mm-hmm. We have two parasites that look similar to each other. One is called Clonorchus sinensis, and another one is called Opistorchus viverini.
Wow.
And they almost look the same, okay? When you look at them, the same. And you acquire them by eating raw or undercooked cyprinoid fish, freshwater fish, right? And they locate in your bile duct. And they elicit hyperplasia because they too secrete proline. I'm sure of this because they're related to the fasciola group. And the hypertrophy that occurs in the epithelial cells can sometimes turn into a metastasis. And so you get, essentially what you get is a cancer of the bile duct. And people die from it. And, it's long-term exposure that results in this, okay? So we all know that the various stages of cancers can sometimes take a lifetime to achieve. So Clonorchus sinensis and Opistorchus viverini has been associated which was the parasite for the schistosomes associated with metastases?
Onchocerca.
No.
Leishmania.
No.
You're talking about metastases.
Schistosomes.
I don't remember.
Schistosoma hematobium.
Okay.
You can get a squamous cell epithelioma developing in the base of the bladder. All right, but there's a co-carcinogen we think that's associated with this process. We don't know whether there's a co-carcinogen associated with this or not.
Now, can the parasite itself kill you by damaging the tissue?
Fasciola can.
Liver damage, right?
Liver failure with enough of these things. Imagine a fluke that's maybe the size of, when you touch your finger to the tip of your yeah, that's how big it is. Take your forefinger and your thumb and put it together. That's how big these things are. Imagine 100 of them.
Yeah, that would hurt. So that would take a long time to build to that point.
Not necessarily. I mean, you could eat them all in one meal.
Okay. Now,
if you're very unlucky.
Is it correct that the vast majority of people infected with these are fine for many years?
They're silent infections. They call them silent infections. And so the answer is yes.
Is anyone ever diagnosed at some point for having symptoms and going in?
Of course. There's tons of clinical cases of these things, particularly Paragonymous, where you get a lot of coughing of blood as the result of these things.
You said, by the way, you could cough out the adults, the eggs.
Eggs.
And then how do you infect someone who inhales your cough?
No.
Just environmental contamination?
No.
How?
No, remember, you need a snail here.
So you cough by water and it goes in the water? Is that realistic?
No, but you're swallowing a lot of these eggs. You're coughing and swallowing.
And they're resistant to your
they go all the way through.
Intestinal tract.
They don't hatch or anything else.
So if someone shows up with this and you diagnose it, what do you do?
That's a very good question, Vince. What do you do?
Ivermectin?
No, it's not effective at all here.
Penicillin?
No.
Acyclovir?
No. Praziquantel is probably the best drug you can give here.
Praziquantel.
And there's a benzimidazole derivative called, and I don't want to make the mistake of this, so I actually printed it out. Mm-hmm. There's another drug called, I'm reading down my list of my cheat sheet. Printed this on purpose. Here it is. Triclobendazole.
It's in your book.
Oh, good. So praziquantel and triclobendazole are two drugs that seem to work very well for most, if not all, of these flukes. But there's some danger, of course, of killing a very large organism that sits in your liver.
Yeah, because then it's stuck there, and then what happens?
It degenerates, and a bunch of other things happen. You get a fibrotic lesion created around each one of these dead flukes, right?
It's problematic.
And if it's in your brain, then I think.
How do you diagnose it, by the way?
Well, you could actually do a CAT scan on this one and see it.
In the liver case.
Yeah.
Do you see, how about in saliva? Could you see eggs in the saliva?
You could if there were, if it was from Paragonymous, you could take a sputum sample and check it out. But the most common way of diagnosing these is by the stool examination for eggs.
Okay. It also says there's a skin test with antigen.
Some of them have further developments because of their widespread distribution. So there are some people interested in developing a larger kind of test that would be easier to run that would not require a laboratory examination.
Why can't our immune system get rid of these? Are they too big?
Well, no, it's not that they're too big. I think that these things secrete a whole bunch of bioactive molecules that interfere with our immune systems, and we're totally unaware of most of those mechanisms.
Nobody studies these.
There's no money.
Could you grow them in some
Good question.
way to study them?
You'd think so because of their habitation requirements in the field. At least you could probably get this thing to grow in invertebrate tissue cultures for the sporocyst stages, and I'm not sure if you could get a Siccaria, all right, to transform.
Yeah. That'd be the
or
problem.
not. And a Metasiccaria, I'm not sure of the details of how that would work. You see what the Paragonymous Metasiccaria looks like. It's a totally circular thing.
Yeah.
You wouldn't even know what you're looking at if you looked at that on a piece of crab meat. And yet, that's going to give you a lung fluke. Again, pretty good sized thing, maybe 1.5 inches, 2 inches long.
All right, I have one more question for you.
Please.
What do we do about this?
What do we do? Well, Vince, there's one simple answer here that's unfortunately very difficult.
Don't eat raw freshwater fish.
That's one. Number two, in places where there are not a lot of animals running around, for instance, you could put fencing around your fish farms.
Yeah, keep out the animals, right?
And then not use human feces as fertilizer. If you did that simple act, you could also prevent this from happening. Aren't you glad, for instance, that freshwater shrimp are not one of the food items mentioned in these articles?
So they're not infected by these.
We eat a lot of freshwater shrimp in this country.
Where do they come from?
Thailand.
But they're not infected by these flukes.
Vietnam. I didn't say that. I'm just saying that we haven't identified any yet.
You think there might be flukes there?
I would love to know that answer. And if anyone out there is listening that knows that answer.
But you could cook the shrimp and you're fine.
Yeah. Anything of these that you cook or even freeze, you could kill them.
You know, Dickson, I'm having pasta. That's it.
No, you could have more than that, Vince. Come on.
Freshwater fish. All right, so if you farmed fish in a vertical farm, you could get around all this because you wouldn't have animals.
Of course you could. You could control everything. Around. Remember, outdoors you control nothing. Indoors you control everything.
Is that the philosophy of the vertical farm?
That is the philosophy.
You're a control freak, huh?
You betcha. In fact, Vince, I just learned recently, like yesterday, that there's one going up in Holland, which is actually underground, 3 stories. So is that a vertical farm or is that an inverted vertical farm?
No, it's not going up either. It's going down.
It's going down.
Neat.
It's actually going down for 3 stories of hydroponically grown plants with grow lights. And these guys figured out the energy budget and they can actually make this happen. I think that's very exciting. They're called Plant Lab, all one word, in case anybody out there wants to check it out.
Yeah, they're online.
Lab. Yep.
All right, Dickson, do you need to go to Brooklyn now?
I believe I do.
You want to wrap it up?
I believe I will. There's tons of details here that I've left out, and everybody knows that's familiar with this subject. I mean, every one of these deserves its own attention span that goes beyond just saying, well, you pass the eggs into the freshwater environment, a snail picks it up, it either eats it or gets penetrated by it. The sporocyst forms the daughter sporocyst, forms the cercaria, forms the metacercaria, then you eat it. That's the general pattern, but each one of these has remarkable stories associated with it. Like the woman that sits on the side of the chestnut harvesting area and cracks open the nuts with her teeth and discards the shells and puts the finished product into a basket for people to consume. The inner part of a chestnut is fine. The outer part is how she caught Phaseolopsis. Just a simple act of harvesting and of putting something in your mouth that hasn't been cooked. And is there a surface sterilant that you could imagine, dip them in that first, kill this, and then you can do that?
Mm-hmm.
Like Clorox or something, maybe, but that would be, surface sterilization of food items is a very common practice in some areas, by the way.
You could gamma irradiate.
It's a lot of gamma radiation you're talking here.
Yeah.
These things are really resistant. Oh, my. We tried to do that with trichinella in meat. And you should see how much radiation they
need require.
A lot. Oh, my God, yes.
We were talking about that on TWiP recently because there's been an outbreak of salmonella on cantaloupes.
See?
And they're talking about gamma irradiation to get rid of that.
Yeah.
They come from places where the water used to wash the melons is contaminated.
Bingo. There's some salmonella species that can actually penetrate the fruit, too.
Yeah, we were also talking about that, right?
Tomatoes, in particular, from Mexico, are a good source for salmonella.
But then when you cut it open, you should be able to see some pulpiness, right?
Not really.
No?
No, it's difficult. This is a very tricky area. I learned all this from a guy that was the head of the Food Safety Board for the state of Minnesota, which had the best food inspection system in the world.
Who inspects food for the U.S.?
That's a good question. The United States Department of Agriculture.
Okay. Not the FDA, right?
No, they're Food and Drug Administration. They set down regulations with regards to a lot of things, but this is not one of them.
Okay.
USDA does it, but it's a big, very, very, very big job, and it's impossible to inspect everything. And so you just have to depend upon habits of cleanliness in order to carry the day.
I have a few short emails I could read. Is that okay with you?
Sure.
I'll not read the longer ones.
No, I'm not in a hurry, Vince.
You're not in a hurry?
I told them 1 o'clock,
okay.
So I think I can be there by 1.
Let's do a couple. First one is from our friend Jim in Virginia. You both have probably seen this already, but for TWiP and TWiM listeners, fighting malaria with transgenic fungi is the first item in the 41-minute podcast, February 25th from Science Magazine. This sounds both fascinating as another means of dealing with animal and plant pathogens, and frightening from my understanding that fungi, at least as a human pathogen, are difficult to control. Could such fungi be transferred to humans, and how could that be tested? Did you know about this, Dickson?
Yeah, I do. That's not a typical fungus, and we've been exposed to those fungi for millennia without any effect on us whatsoever, so I'm not worried about that part.
I think we should talk about this on TWiM, since it involves a fungus.
Yeah. When I was at Notre Dame, by the way, and I did a little bit of work with Aedes aegypti, which is the yellow fever
mosquito.
Yeah. The larvae of that organism are subjected to an algae, which actually attaches to the larvae and kills it.
Right.
So I was fascinated by how many enemies everything has out there.
I think that podcast also talks about a new palm-sized NMR device. Man, it just keeps getting better and better.
Yeah, it does.
And then he goes on to talk about some of his experiences. I've talked about visiting Weed, California last summer.
Yes.
As a kid, Jim writes, I lived a few miles south of Weed in the logging area of Mount Shasta next to the mountain, as well as in a railroad town of Dunsmuir. A few more miles south, you can also see Black Butte, just north of the town of Mount Shasta along the interstate. Black Butte is a cinder cone about 2,000 feet high with a path on the backside that's visible with Google Earth that used to lead to a U.S. Forest Service fire watch station that was continuously manned. I think it took us kids a couple of hours to climb the path. If you get back that way in the summer, have the time and want the exercise to climb up Black Butte would be a pleasant adventure. Mind the altitude, though. The starting point is 4,000 feet. For Dickson, a couple of miles west of Shasta up in the Cascades is a small mountain lake, almost a pond, maybe 20 acres in size, with 2-foot-long rainbow trout that appear to float in midair. The water was so clear.
Wow.
The lake only supported a few of these beauties, and they were impossible to catch. The lake name, which you can locate with Google Earth, was either Porcupine or Toad Lake. Porcupine seems to be the one, but I don't see any logging roads to it, as appears to exist for Toad Lake, and the one with the fish could be reached by road. Well, you had to walk to the other. That was 50 years back, though. There's a webcam for Mount Shasta. He gives a link with several days' worth of time-lapse viewing that I look at. The same site provides temperature data. It was interesting as a kid to have a mountain in your backyard. I bet.
No.
I had a city in my backyard.
You know, Vince, there's a cinder cone in Newark.
You're kidding.
No. You pass it every time you take Amtrak down to Washington.
What's it called?
It isn't called anything, but you've seen it. I know everyone has seen it.
Oh,
this is just it looks like a big rock rubble.
I'll have to look it up.
And no one knows what it was, except a friend of mine told me. He says, "You see that over there? That's a cinder cone."
So that's from a volcano.
Yeah, and all of the Palisades are all basaltic rock.
Yes, that I knew. The next one's from Eric. Thanks for reading my email. The sync trick I referred to was the synchronous mouse infection that Dickson describes at 15 minutes 20 seconds into TWiP number 5. Okay, because on a previous TWiP, we read his email. We didn't know what he was talking about by sync. The synchronous mouse infection that you talked about, remember? With what parasite?
Well, I can do this with Trichinella.
TWiP number 5 would have been.
Must have been Trichinella.
Let's look. Just quickly, Microbe World.
Aren't they great? I love worms.
TWiP 5 was the Trichinella. Mm-hmm. You bet. So that was a synchronous infection with Trichinella. It's wonderful that Dickson shares this with the new researchers coming up. It's a bit like a magic trick. Difficult at first to see how it's done, but much easier once you know the technique. In rooting out computer bugs, if you can repeat the problem, you can always find its cause. The sync trick is like repeating the problem many times in parallel, so you only need to run the experiment one time. And if you have to destroy a cell to see what stage it's at, you have many others still running at the same pace. Love it. Parallels between science and computing.
Absolutely.
Jim writes again, our friend from Virginia. This is for Dick in the informatics realm, in case you've not kept up with the different presentations. It is a bunch of TED Talks by this fellow. Well, this is Gapminder.
Oh, I've seen him. No, I've actually seen him. He's great.
And Hans Rosling.
Yeah, he's fantastic. He's amazing.
He's done a bunch of TED Talks.
Yep. In fact, I think he was at one of mine. One of mine. I only gave one big TED Talk in 2009. This was 2008, I think he did it. But
trends, world's most important trends, CO2 emissions since 1820.
Yeah, the guy is fantastic, and he does this in a visual way, so you can't miss the data. Oh, this is very cool.
So this is sort of like the other fellow who does the visual representation of data.
Correct.
What's his name?
It's Edward Tufte.
Edward Tufte. It takes a while to load this.
Yeah, it's big. It's done to scale too, so I mean you can get relative proportions, etc. And it's a movie, so it's depressing to watch it.
I see. You play it, and you can see how CO2 emissions have gone up. I presume.
Of course.
All right, the next one is from Stephen. Isaac Asimov was a biochemist, not a physicist.
Mea culpa.
As mentioned in episode 23.
Yeah, we take it all back and totally agree with you. From start to finish.
He did write many popular science books explaining physics and astronomy. I have 3 or 4 of those, so it is an easy mistake to make. Yeah, see, he gave you a little slack.
That's very nice of him. But, I do make mistakes, just like everybody else, and I don't take offense at being corrected because I want everybody to know what the real truth is.
The next one is from Raphael. our friend in the Philippines. I've been doing some transcribing in my free time on your TWiP's hookworm episode for my blog site.
Yeah.
And it seems that Dr. Dick made an error on mentioning the term hematuria for the dark brown colored stools found in patients with intestinal bleeding. Hematuria is meant for blood in the urine. What Dr. Dick was supposed to mean was melena. And contrasting this to the word hematochezia for the bright red stools, undigested fresh blood. Raphael's a physician.
I sit corrected.
You sit corrected. If you're interested to
hematuria is definitely urine.
If you're interested to note the error, it's around 39 minutes and 55 seconds of the recording time.
I bet you we mentioned it, though, correctly when we talked about Schistosoma hematobium, because there is hematuria associated with that one.
Well, I'm sure Dr. Raphael will be on top of it.
I hope so.
And will let us know. Thank you, Raphael. And our last one is from Judy, a high school teacher in California. Thanks for the great podcast. I learn a science, history, and a bit of politics from you and love the unexpected connections you make. Abe should have given a medal to the hookworm as the supreme undercover agent. I am a high school teacher, and I try to teach my students to think about things. And one of the things I've been pushing is that virtually every decision or action has a potential positive and a potential negative. You weigh both sides to make your decision. In that vein, I have to ask the other side. When you talk about parasites, you talk about them in relation to human diseases, and rightly so. The goal is to rid the world of them to protect people. My question, do they have no other function in the larger ecosystem? Can we rid the world of onchocerca or hookworm or malaria for that matter and have no effect on the larger ecosystem? If these are truly a single food chain rather than being part of a food web, I will use them in class as an example. Right now, I tell kids that almost all organisms are part of a food web, and so we need to look carefully before to bulldoze or plow or pave or overfish.
Hear, hear.
Thanks for keeping me learning and interested, and for the best professional development I have ever gotten. I strive to teach as well as the two of you.
Wow.
That's a compliment, Dickson.
That's Vince, and for you too.
No, it's for you.
No, okay, but my answer to her is that that's a fabulous question to ask. And for the parasitic infections that are less host-specific, say, like, for instance, on TWiP, we've discussed West Nile virus, okay, and it's very promiscuous. And if you look at it, it infects invertebrates, cold-blooded vertebrates, and warm-blooded vertebrates. What would be the effect if you got rid of the West Nile virus? I think in terms of some ecological situations, it's important for population control, and therefore you don't have a lot of these or a lot of those because they're being kept in check by their parasites. With a host-specific parasite, like malaria, in the case of Plasmodium falciparum, if you got rid of Plasmodium falciparum, the only organism to benefit or suffer from that would be humans. Mm-hmm. So I would say it would be a great thing if you could get rid of the human-specific parasites, unless you thought that controlling human populations by the death rate in parasitism is a good thing, or is a neutral thing. And in fact, I think the best way to present life to your students is life is neutral. The earth and evolutionary process of progress and process is neutral. You can't make a decision if you take the side of the lion trying to feed her poor little hungry cubs and watch it take down a zebra and saying, good, she got the zebra, and those little kids can now grow up to be killers. Or if you take the side of the zebra and say, oh, look what that poor zebra how could that vicious... okay, so those are two completely unacceptable views in terms of evolution. But in terms of us, because we're so species-specific, and because we care a lot about everybody else, we tend to take the side of humans almost every time.
Sure.
So it's a great question, though, because are any of these parasites that infect us and others too involved in population control, which may be a very good thing in the long run? So can we specifically eliminate the parasite from our environment and not from everybody else's? Like, for instance, all these flukes that we just talked about. These are flukes that infect other animals too. And do dogs suffer from this, or do cats suffer from this, or do the fish, or, any of those questions are valid. the answer is we don't know enough about these things to really make a decision.
I mean, your point has always been that they're involved in population control.
Yeah, in general, parasitism is a ecological concept that involves host-specific directed population control mechanisms. That's exactly right. And the ecologists have come to accept this, and now they think we've been neglecting parasitism all of our lives because we've considered all these other things first. But now we have to include it as a major concept because it drives populations towards or away from being balanced with the rest of nature.
So are they part of the food web?
Which one? Tell me which
all
one.
All the parasites.
Not all of them. You can't say all of them. You have to say certain ones.
Okay, because she said if they're truly a single food chain.
Well, it depends on the parasite, remember? Because if it's a specific parasite just to humans, the answer is no.
Okay.
And if it's a parasite that has promiscuity associated with its host range, the answer is probably yes.
Okay.
And that's why I would divide it off. If we wiped out all of the human parasites that were only specific for humans, then our species would be uncontrolled by parasitic infections. But we control our populations in
other ways, sure.
It's all economic, basically.
Right.
I mean, so here comes the politics.
So you're dividing the human parasites, specific parasites, from everything else.
Yes, because if the host specificity for humans is not just us, but other things too, then other things may be controlled by it. And it's a fascinating question that has no specific examples that I could actually give you that you say, well, you go to that literature and you can check that out. All right. It's not going to work like that, unfortunately, because they receive too little attention from an ecological standpoint. But they should get lots more.
All right, Dickson. Thank you very much.
You're welcome, Vince.
microbeworld.org/TWiP or on iTunes is where you can find This Week in Parasitism. Please subscribe if you can in iTunes and leave a comment on the front to keep us on the front page of the medicine podcast section. There's also a app that you can use to listen to TWiP on your iPhone or Android device. You can find it at microbeworld.org/app. And as always, send us your questions and comments. We love them. TWiP@twiv.tv. Dixon Despommier, of course, is at Racaniello.org, medicalecology.com, and verticalfarm.com. Thank you, Dixon, as usual.
Welcome, Vince.
And I am at virology.ws. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.